Okay, well, welcome to the blood lab. You just finished the lab exam. I hope that went well. What do you think? Lab exam go okay?
Just okay.
Just okay? Better than the first one?
Mm-hmm.
First one was a little rough, so I'm seeing happy faces, right? Happier faces. I just take a look at them. They're looking pretty good, so I'm pleased. Hopefully the scientific method part was very straightforward, right? Kind of a gift. What we're going to be doing tonight is taking a look at a blood lab. This will be our only blood lab of the semester. And as I was saying, in your supplement, the PowerPoint for this did not make it into your packet because without them being in color, they're not very useful. And we just pulled them out because they were just big black blobs that weren't helpful for you. You will find this PowerPoint, though, posted under Week 6, Lab 7 on blood. Also under here, there's a flow chart that you have in your supplement. If you wanted to have another copy of that, that is here. There is the presentation that I'm giving, and then the images that you're going to be, I'll put this up later on, but this is the page that you have, I think on page 170 in color, okay? So that's also online for you, and that may be helpful for you because identifying these white blood cells is very much dependent upon color. So having black and white blobs is not very helpful. So with that in mind, let me just jump over to the presentation and walk you through this. Now, for those who were in lecture, some of this will be a nice little review. Those who weren't, though, I did tell them that I was going to specifically go through some of this in lab. So I did jump over a little bit of stuff in lecture, knowing that I would spend a little bit more time with you this evening. Now, after we do this presentation on the iPad and get this lecture recorded, then we will be going down to room 344, and that is a wet room where we'll actually be doing a little bit of work that requires a sink. So we'll be going down to yet another room this evening as we're being shuffled around. Now, this is, let's switch our minds, right? Those who were in lecture have already kind of done that switch, but let's switch our mind. Blood will be the beginning of your third lab exam, and blood will be the beginning of your next lecture exam. So this is a new beginning. It's really the second half of the course as we pick up right here. If you were to take a drop of blood and put it on a microscope slide and stain it, you would see not only red blood cells, but you would also see a host of white blood cells that will stain differently from one another. And those are the white blood cells that we see on this image. Now, the cells that you know the best would be the red blood cells, right? And there's 1, 2, 3, 4, 5 red blood cells here. We saw them in 105. We know that under different solution conditions, they can swell in a hypotonic environment. We know that in a hypertonic environment, they can shrivel down or crenate. We know they are kind of able to change their shape. What you also see are a bunch of larger cells, and these are the collection of white blood cells. And there's 5 different types, and I have slides and more information. You really can't read this. But these larger cells, notice they're different. They have a slightly different set of combinations of colors and textures, and we'll learn about those. And then finally, there are the little blebs. Those little blebs are the platelets. Collectively, all of these cellular and cellular structures are called the formed elements. And on average, in this room, if I took some of each of your blood, about 45% of your blood by volume would be composed of the formed elements. The other 55%, the remaining volume, would be composed of the plasma, the liquid portion of the blood. Now, red blood cells. What's their size? What's their width across is 7.5 micrometers. And in lecture, I mentioned also 8. So there's a little bit of discrepancy, but typically you'll see 7.5 as the standard or 8 micrometers. Very, very small. And the middle picture is a scanning electron micrograph of the head of a pin, of a sharp pin. And there you can see these little red blood cells, right? Each of these little tiny, tiny red blood cells. So it kind of starts to give you a perspective as to the size of these cells. On the right is an electron micrograph showing you red blood cells. Now, the shape of red blood cells, the name for this shape is biconcave. That double indentation, biconcave. And that biconcave shape actually increases the surface area of this cell. And you can see in this picture, let me change colors here. This is a capillary, okay? And a capillary, recall, is about 8 to 10 microns across. And red blood cells are about 7.5 to 8. So it tells you that the squeeze right through a capillary is rather significant, and that these red blood cells have to sort of shimmy their way through and bend and squeeze their way through those capillaries. The numbers here at the top, in a male, 4.6 to 6.2 times 10 to the 6 million red blood cells per microliter of blood. Recall that a microliter of blood is the same as a cubic millimeter. So imagine making a little cube with every face of that cube being a millimeter in size. Very, very small little cube. And filling it up, that would be 1 microliter of blood. And in that little microscopic drop of blood, there are between 4 and 6, roughly, million red blood cells. In a male, and in women, 4 to 5. So a smaller number, right? A lower number of red blood cells. Now that lower number of red blood cells would equate to what? What are we going to be doing tonight? If you looked, and what would Dad talk about in lecture if you were there? When we take the blood and we separate it out, the more red blood cells, the higher one's hematocrit, right? And hemato, we'll be doing a hematocrit tonight. And hemato means blood, and crit means to separate. So we're going to separate the blood into its components. So we're going to take some blood. The blood we have tonight are some animal friends. We got goat, sheep, and horse. And we'll be taking some of these animal bloods and actually doing a hematocrit and watching the blood separate out and looking at the percentage of blood composed of red blood cells versus the plasma. These numbers are also listed for you. I'm going to give you a page number here. Stacy, give me a hand here. If you turn over to, I'm going to find it. Well, hold on. They're in here. These numbers are in here, but I want you to go right now over to the hematocrit number. So go to the top of page 175. Top of page 175, and there you'll see in bold some numbers. Again, there's some numbers in this lab that you need to learn, and these numbers are going to help you in the lab as well as in lecture. So at the very top, you see the hematocrit values. Now, hematocrit values are given to you as percent. And we'll talk more about the hematocrit when we get to the other room. But what does it say? The average male has a hematocrit of what? 40.
Oh, 48.
That's the average, right? 48, with a range of 45 to 52. So I just remember kind of hovering around 50, right? Hovering around 50%. And then women have a lower hematocrit because, look at the numbers. They have fewer red blood cells per drop of blood. And so their hematocrit is in the high 30s up to what? Around 48. Do they cross over? They definitely cross over, right? And what really drives the male hematocrit to be higher is that testosterone does, by its very nature, drive up the amount of red blood cells. And that testosterone also drives up the amount of muscle. So the muscle and the testosterone and the red blood cells all kind of go together. So a little bit about red blood cells. If you took a smear of blood, you would see mostly just red blood cells. Red blood cells, as I mentioned in lecture, account for about 99.9% of all of the formed elements. So mostly what you're going to see are red blood cells. But you will see occasional larger cells. Now, these cells are not naturally their other colors. These colors are going to be enhanced by dyes or stains that are added to the blood. But red blood cells are themselves naturally red, right? And what makes red blood cells naturally red is the the pigment inside called hemoglobin. And hemoglobin is naturally bright red. So red blood cells don't need a stain to see the color, but the other white blood cells, in order to see them the way we're going to see them, does require that you add some stain to them. Here, we're seeing one white blood cell. This happens to be a lymphocyte. We'll learn how to identify it in a few minutes. And what you see is that this cell is bigger than a white blood cell. The other thing I see, if I look very, very carefully throughout this image, I see a few little fragments, and those little fragments would be platelets, right? So we see those, again, relatively rare in comparison to the number of red blood cells. So that's enough about red blood cells. Now let's dive into white blood cells. We're going to have fun with this tonight. I think this is a fun lab. Now, white blood cells are all about your immune system. Red blood cells, their job is pretty much exclusively to carry oxygen and a little bit of CO2. White blood cells, 5 different types, and then some subtypes of those, their job is to help with your immune system fighting off disease and pathogens. There are 2 main groups of white blood cells, granulocytes and agranulocytes. So I might say types or classes or groups. Okay, any of those words would be the same thing. And the granulocytes have, by their very nature, some granular appearance. These are internally in the cytoplasm vesicles, right? They're like little vesicles filled with enzymes and proteins. Neutrophils, eosinophils, and basophils all have these granules, whereas lymphocytes and monocytes do not. So they are the agranulocytes. Okay, so the granulocytes and the agranulocytes. Now the term neutrophil, basophil, eosinophil, what does the last 5 letters mean? Phils. If something is philic or phils, hydrophobic, hydrophilic, what does philic mean? Love of or an affinity for. These cells were named by their affinity for the dyes. So the dyes that are used, there's no affinity. The neutrophil has no affinity. It's neutral. So it's the neutrophil. The eosinophil has an affinity for a love of the dye eosin. And that eosin is a red dye. And it gives these cells a very bright red appearance. And I'll show that to you. And then the basophils have an affinity for the basic dye, which gives these cells a black-blue appearance. The lymphocytes and monocytes also pick up color, but they don't have the granules. So let's take a look at these different cells. These pictures that I'm showing you are fragments from the next page over, right? Okay. So if you want to take a moment, I know I have it for you on Blackboard in color, but if you turned over to page 170, you might want to make a couple just notes about what color these slides are. And then as I give you details, you can be filling in that table, that pre-lab table. Now, these are both examples of neutrophils. The most common neutrophils are called segmented neutrophils because the nucleus appears to be in multiple segments. So you'll hear them called segs or segmented neutrophils. These are mature neutrophils. And they account for 50 to 65% of all of your white blood cells. So they're by far the most numerous, right? Neutrophil, numerous, neutrophil, neutral in color. And most of them, again, are segmented. Now, there is another type. Actually, it's the same thing. These are called banded neutrophils. That's what's shown on the bottom. And if you look, the nucleus is not broken up into segments, but appears to be one solid band. These neutrophils are young. Okay, so this is a young neutrophil. This is, I won't call it immature, but newly formed. Let's just call it a newly formed neutrophil. They're going to account for none at all to 5%. So that means collectively, what can neutrophils represent? Collectively, neutrophils will be 50 to 70. Okay? So collectively 50 to 70 when you add the segmented to the banded neutrophils. Neutrophils are the type of white blood cell specifically designed to fight off bacteria. So these are your bacteria fighting cells. When a bacterium invades your body, these cells are drawn toward that bacteria. I mentioned today the word chemotaxis in lecture, that bacteria themselves release chemicals that attract these cells to them. And then these cells will, quote, degranulate. They will release their granules. And those granules have within them antibacterial factors designed to destroy the bacteria. So these are your bacterial cells. Now, on the bottom are your banded neutrophils. If you had a new infection, a new acute bacterial infection, you would have a higher number of the banded neutrophils because these cells, right, are newly formed. So your body has newly produced more neutrophils to fight off that infection. I mentioned in lecture tonight that platelets and red blood cells do not divide, right? They are not true cells. And so they are produced and are destroyed and constantly remade. Whereas neutrophils can divide. They are true cells. They have a nucleus. They have a metabolism. And when there's an infection, more of these cells are made in response to that infection. And so you would have more of these banded neutrophils in your blood if you had an acute infection. The second leukocyte to take a look at are the eosinophils. Now, notice that the granules in this cell are very bright red. Okay, they'll go from being bright red to hot pink to salmon to orangey, but always quite distinctive and brightly colored. And it's that eosin dye that picks up, or those granules pick up this eosin dye, hence their name eosinophils. They will only account for 2 to 4% of your white blood cells. So you can drop that 2 to 4 in your front page. And these cells are designed to destroy parasites. So if a person has a parasitic infection, a worm or some sort of tapeworm, then these are the cells that your body will recruit to specifically destroy the parasitic infection. The bottom one, basophils, they don't have a bright red granular structures, but instead have a dark bluish black granules. Those granules release histamine. These cells are the rarest of your white blood cells. They're going to account for less than 1%. So basophils, blue-black, numbers in the basement, right? All the B's. So the basement numbers, blue-black granules, basophils. And they release histamine. So if you were having an allergic response, your body would be making these basophils and releasing histamine. And what does your body do then in response to this histamine? You go to Walgreens to get antihistamines, right? To stop it. So what's happening to you? What does this histamine stuff do? It makes your eyes water, makes you sneeze, right? So your body is trying to get rid of whatever it is that's giving it a hassle. So if you're having an allergic response, having hay fever, some sort of allergic response, you're going to have a higher number of these basophils. The second most numerous type of white blood cell are the lymphocytes. The lymphocytes are going to account for 25 to 33%. I do believe that the lab in the lecture it said 20 to 40. Okay, so there might be a little difference in discrepancies in these normal ranges. But 25 to 33% is what I have here. These are the smallest of the white blood cells. They are not a whole lot bigger than red blood cells, but what makes them distinctive is that they have a very large, round, solid nucleus that almost takes up the entire cytoplasm. So it kind of looks like an eyeball looking at you. Okay, they're the smallest, but a very distinctive nucleus and no granules, right? Nothing that we see here is granules. Now, these lymphocytes come in two basic flavors, T cells and B cells. right? So you'll hear about T lymphocytes and B lymphocytes. That's a conversation we'll have a little bit more of when we get to the lymphatic system. And if you take microbiology, you'll have a nice dose of the immune system and the T cells and B cells. On the right is an atypical lymphocyte. I'll deal with that a little bit more later, but I should say lymphocytes are the cells that are going to be specifically attacking viral infections. Okay? So if you have a viral infection, it's the lymphocytes that are going to be specifically recruited to fight off that virus. Sometimes when a lymphocyte gets infected by the virus, it becomes an atypical lymphocyte. So if you look on the bottom right, that cell certainly has some very strange looking amorphous shape to it, right? It's no longer a nice round cell. So something has definitely kind of overtaken that cell. We'll see that in one of our case studies tonight when you're in the lab. So that is the atypical lymphocyte. The last one, the fifth one, are the monocytes. They are going to be most distinctive for their size. I always remember that monocytes are monstrous in their size. They're 5 to 6 times bigger than erythrocytes. They could be nearly twice as big as the other white blood cells. Their nucleus does tend to be horseshoe-shaped, but that's not really going to be your differentiation. It's going to be the incredible size of these cells. These cells become macrophages. So these are the cells that can gobble up things. They account for 3 to 8%. 3 to 8% of your white blood cells. So that's a quick run through these different cells. Anything to fill in, anything you're not sure about, based upon what you're seeing on your tables? Platelets haven't talked about yet. Give me a second on platelets.
The function of monocyte.
It's a macrophage, so they can start gobbling things up, right? So in lecture tonight, I mentioned there's actually 3 cells that can be phagocytic, and the monocytes are one. The other one is the neutrophil and eosinophil, right? So the monocyte, the neutrophil, and the eosinophil are all capable of phagocytosis, but this is the big one, right? This is the big chewer. Let me give you a couple other words while I'm thinking about it. So somewhere in your supplement, it tells you, and I mentioned it in lecture tonight as well, that a normal number of white blood cells should be 5,000 to 10,000 white blood cells per microliter, right? That would be a normal range of white blood cells in your blood, with an average of around 7,000 or 8,000. If you had less than 5,000 white blood cells, a low number, right? Unusually low, then you would be considered to have leukopenia. You may recognize penia as a deficiency in. So leukopenia would be a low number, a deficiency in your white blood cells, so less than 5,000. If you had a higher number than 10,000, if you had an abundance of white blood cells, I'm going to put an up arrow. If you had too many white blood cells, we would say that you have leukocytosis. Okay? So osis, an abnormal condition of many white cells, right? Leukocytosis. What would be your symptoms if you had leukopenia? Lots of infections, right? You'd be getting sick easily. What if you had leukocytosis? You may already be sick, right? The reason you have too many white cells is that your body is actively fighting off an infection. Okay. So we'll just keep it simple for now. Now, let me also put a couple more words up here. Let's try this one. We'll go to the chalkboard. Okay. I'll pretend I'm Khan Academy now. Okay, we'll go with yellow. Okay, here we go. So if you had red blood cells, so give me the numbers again. A normal male, what should their hematocrit be? I don't memorize these ranges because they kind of move around, but what does it say? Not the raw number of cells, but the hematocrit, the percentage values? 40, that was the average. Range? 45 to 52, right? And that's percent. That's what your HCT should be. That's your hematocrit. That's for males. And for females, it was 38 to 40, 37? 37. See, Khan Academy, they never make mistakes. 37 to what, 40? 48. 48, thank you. So that's what the normal range should be for the hematocrit. What if your hematocrit is too low? You would be diagnosed with low hematocrit, anemia. Right? You've got anemia. You've got too few red blood cells to get oxygen to your tissues. If you had a high hematocrit over the number, what condition do you have now? Polycythemia, good. So poly, and all that means is emia, condition of the blood, too many cells, right? And because red cells are by far the most abundant, polycythemia is referring just to the red blood cells. Now, how would you get polycythemia? Give me a suggestion as to how you might have too many red blood cells, or at least have more than the average hematocrit. Yeah, right. If you were to suddenly climb to a higher altitude, and at a higher altitude, your body will respond by making more red blood cells, then you may, for a short while, be polycythemic. The problem with being polycythemic, long term, short term it's okay, but long term is that it makes your blood thicker because you have more red blood cells. So it makes your blood more viscous and could potentially make your blood more difficult to travel through capillaries. Slow it down a little bit, make it difficult. And so, the number of red blood cells is perfectly designed so that blood flows without any consequence. So I do want you to know those four terms: leukocytosis, leukopenia, anemia, and polycythemia, and how they're connected one to another. The last thing, and I didn't give you numbers for this yet, I'm going to give them to you right now on platelets so that you have your front table filled out. So for platelets, now I definitely feel like Khan Academy. I'm going to go with pink. Okay, so for platelets, the number of platelets should be, I just can't write as well as I do. Platelets, 130,000, up to as much as 500,000, but that's really high, but you'll see that as a range. I think that's what the Martini book gives us in the lecture. So 130,000 to 500,000, this is what? Platelets per microliter of blood. So all of these are per microliter of blood. If you had a decreased number of platelets, what would your condition be? What's the other name for platelets? Thrombocytes. Now, the problem with that word, right, is that platelets are not cells, but they have been historically called thrombocytes. You can't even read that, can you? They've been called thrombocytes, and thrombocytopenia, therefore, would be a deficiency in these in these platelets. What would your symptoms be? Can't clot, bruising easily, lots of bruises, right under the skin. You'll see a person, if they have low platelets, every time you bang your hand, they get bruised. That's because their platelets are low. Okay, so now you've got all the numbers, I think, for page 167. Is there anything that you're missing? Right, platelets function to help with blood clotting. They're just very small. They're about the quarter of the size of a red blood cell, so they're very, very small. So they're not going to be hard to recognize.
And they're formed from megakaryocytes.
And yeah, the platelets are formed from megakaryocytes, and that is something I briefly mentioned in lecture today. So that is sort of the introduction to the cells. When you go to the doctor, they're going to do a complete blood count. And one of the things they're going to do is draw your blood and look for a white blood cell differential. Now, this is done nowadays by machines. In the old days, however, they would draw your blood, and literally a technician would be looking through the microscope, and within a certain field of view on a little instrument on the microscope, they would be counting the number of monocytes, neutrophils, basophils, eosinophils, neutrophils, and coming up with a percentage of each and reporting back about if that was normal or not normal. We're going to figure out tonight, we know the normal ranges, I've told you, we'll learn them, and we'll figure out what that means. But can you imagine all day just kind of doing counting of cells? It's quite the monotony, but they became phenomenally good at it. Nowadays, this is primarily done by machines. So they take a little bit of blood, it goes in a machine, the cell counter actually does this for us electronically and digitizes the report, and it spits it out very, very quickly. But we're going to learn how to do this kind of in an old-fashioned way so you appreciate and can identify these cells. You would also perhaps do a hematocrit. Now, hematocrits are not done very much anymore. Again, because we have machines that can quickly identify the percentage of your cells that are red blood cells. And so we don't do this as much, but I want you to see this. You'll appreciate it and understand it better by going through this process this evening. So that's the two things we're going to be doing tonight. We're going to be doing white blood cell differentials. We're going to give you some case studies. We'll go to the other room and do that. And you'll be doing a hematocrit with the sheep, goat, and horse blood. And also, we're not doing it, but the machine would also be counting your number of red blood cells. So the hematocrit is reported in what? Hematocrits are what units? Percent, right? So hematocrit is a percentage, always a percentage. Again, average in the room, 48. Men hovering around 50%. Women in the mid to low 40s. That's a percentage. Whereas counts would be in the millions. So 4.8 to 6.2 times 10 to the 6 million, cells, right? So when it says RBC count, I'm talking about a raw number of cells. When I'm talking about the hematocrit, I'm talking about a percent. But remember, it's the same thing, right? They're not, well, not the same thing, but they are connected to each other. The more cells you have, the higher your hematocrit value will be. So here's what the hematocrit looks like. Of course, we don't have the animals here, and we're not using human blood. But you normally would draw blood from your vein. And remind me, why do you draw blood from a vein rather than an artery? Less blood pressure, good. Because if you hit an artery, it's going to squirt and make a big mess. And the veins are more superficial, they're easier to get to. So two very good reasons to grab blood from a vein. And we're going to take some blood. We've already got it. The blood we have is guaranteed pathogen-free from a vet service for classroom use. So we don't even have to wear gloves tonight, although we do have gloves in the room if you don't feel comfortable. But this has been certified pathogen-free animal blood. And we'll take a little bit of that blood, we'll put it into a tube, and I'll go through this procedure with you when you get to the other room, and then we'll put it in a centrifuge. It's the last thing we'll do this evening. And then we'll take it out. And we'll simply look at the tube, and let me remind you what this picture shows you and then how to measure this. And then I think will be time to go. After you spin the blood, the red blood cells are all going to pack down to the very bottom of the tube. That is going to represent your hematocrit value. There will be a very thin layer called the buffy coat. The buffy coat is a little interface. It is where the white blood cells and the platelets will pellet after centrifugation. And then everything above it will be the plasma. And the plasma will have the water, mostly, it's about 92% water, have proteins, it'll have hormones, gases, nutrients, waste products. Yes?
If the platelets are smaller than the erythrocytes, why don't they go to the bottom?
Yeah, the question is, it's interesting. The red blood cells pack to the very, very bottom, and that's based on density. Okay? So the pelleting in the tube is based upon density, not upon size, okay, but on density. The red blood cells are just denser. So they go down to the bottom of the tube, and everything else stays on top, okay? It's just know it, but that's why. Now, some books, just to clarify, some books will say that the hematocrit value is equal to the percent of red blood cells. That's how I normally discuss it. right? The number of red blood cells you have, the percentage of it, that's your hematocrit value. Other books will say that the hematocrit is your percent of formed elements, which means your red blood cells plus leukocytes and platelets. But platelets and leukocytes only make up about 0.2%. So the difference is insignificant. So I honestly, if I look this up 5 different times, I'll find at least half of them telling me it's formed elements and half of them telling me that it's the red blood cells. But clinically, it's the same value. It's not going to make a difference. The percentage will not be influenced significantly by that little buffy coat. Okay. So when we do this, we'll look at this, and let me show you, kind of draw out what you will be seeing. You're going to get this tube. So here's your tube. And there will be some blood in here, right? And after we centrifuge this, the blood will be down here. And then above that will be the plasma. And the plasma will be kind of yellowish, kind of a tan yellow color. Now, I'm going to increase the blood. The blood goes up to here, okay? And then the yellow keeps going all the way up. So we take this out of the centrifuge, and now we have to determine our hematocrit value. It's very simple. You take a ruler, and you simply measure the volume, but not in milliliters, but simply in millimeters. Measure the distance that the blood sample is in the tube. So let's say this is where we fill the tube all the way up, and that this is, just for fun, 39 millimeters. Okay, so the entire volume is taking up 39 millimeters in the tube, and that the blood is 13 millimeters. So what is the hematocrit? 13 divided by 39 is? 33%. You recognize as a third, right? 13, 26, 39. So that's 1/3, right? So the red blood cells are making up, it's about 1/3, right? About 1/3 of the volume is red blood cells, right? So this up here would be 26, right? You everybody with me? So that's 26 millimeters of plasma, 13 millimeters of red blood cells. Therefore, 33% is your hematocrit value. If it had been half and half, the hematocrit would be 50%. What we're going to find is that the animals that we're looking at all have hematocrits in the 30%. So we don't have, I don't know if they're males or females or what they're, anything about horse, goat, or sheep hematocrits. I know what our normal values are from human, and that's all that really matters. If, however, I did this and I got a hematocrit of 33%, what can I say about that person, male or female? It's low. They have anemia. That's the diagnosis we'll give them. If I did this hematocrit and it was 40% and it was a female, she's okay. But if I did it and it was 40% as a guy, he is anemic, right? If I did it and it was 50% for a guy, I would say that he is normal. If it was for a female, I'd say she has polycythemia. Okay, so that's what I want you to appreciate. We'll learn how to do the hematocrit, but what's most important is that you can read it and apply these terms, anemia and polycythemia, and in order to apply those terms, you need to understand what the normal ranges are. All right, so you need to know what's normal for male and what's normal for female in order to make that diagnosis. Okay, so that is our little presentation for tonight. So I can't think of anything else I need to tell you right now. We're going to walk over to room 344. We'll go over there together. It's straight out the door and straight ahead, out the door, outside, into the next little hallway. And there I'll meet you, and we'll open up that room, and we'll actually do the counting of white blood cells and do a hematocrit. And we'll still be out of here a little bit early. Okay? Very good.